Hello, today we're going to talk about cellular respiration. Cellular respiration occurs in the mitochondria, and the whole purpose of the mitochondria is to produce ATP. It has three main steps: glycolysis, which actually occurs in the cytosol outside the mitochondria, then the Krebs cycle, which occurs in the matrix, and then the electron transport chain, which occurs on the cristae membrane on the inside. At the end of this, the net outcome from one glucose molecule is 38 ATP. So let's start at the beginning. In the systole, which is the cytosol, same thing, near the mitochondria, a glucose is going to go through a series of chemical reactions. Now, these reactions are the same reactions that occur in a bacteria cell. They don't have a mitochondria, so they can do glycolysis, but that's as much as they can do. So this is what happens. Go to a picture. Okay, first of all, the glucose is going to be broken down in a series of chemical reactions. The names of these molecules are not that important that you remember, excuse me, all of the names, but what happens on the sides is important. The green are the enzyme names that make each of these reactions occur. Again, the green is not that important. So let's take a look at what happens. From one glucose molecule, first thing we have to do is add some energy to get the reaction started. So ATP is going to come in and drop off a phosphate group, giving energy, and then giving off an ADP plus P twice. At the end of that, we have created a glyceraldehyde 3-phosphate. Do you remember during photosynthesis, when we went through the Calvin cycle during photosynthesis, just at the very, very end, I had two G3P molecules that combined to form one glucose molecule. That is this glyceraldehyde 3-phosphate. That's the G3P. So it's the same molecule. We made glucose. Now we're going to break glucose down into that. Then we're going to go through a series of reactions. Okay. During those reactions, an NAD molecule is going to be reduced to an NADH on each side. You go back. See how this forms? Whoop, two sides. Well, both of this is interchangeable, so this one becomes a G3P also. Both sides will be the same. So I got them the same. We're going to start coming down. So I'm going to do the NAD reduced to NADH twice. We're going to create an ATP molecule. Boom, boom, twice. Here's another one. Boom, boom. So during this reaction from here to here, energy is released. And so I'm going to store it as an ATP molecule. Now, what's my net gain? Here I produced 4 ATP molecules, and I made 2 NADHs. Go back up. Here I used 2 ATPs to get started. So what's my net ATP gain? 2. 4 minus 2 is 2. My net gain is 2 ATP, and I've created 2 NADHs. I also created a molecule called pyruvic acid, or pyruvate. Pyruvate then is going to move into the mitochondria. As it moves through the mitochondrial membrane, it goes through a series of reactions called transition. During those reactions, I'm going to give off a carbon dioxide molecule. This is the carbon dioxide that you breathe out when you're breathing. This is where it comes from. And I'm going to reduce another NAD to an NADH plus H. Okay, so I'm going to make another one of those. I'm also going to combine with something called a coenzyme. So let's see what happens here. Give off the carbon dioxide, NAD is going to be reduced to NADH. Here comes a coenzyme that's going to come in and help the next reaction to occur. It combines with the rest of this pyruvate that's left over, which is an acetyl group. So this becomes acetyl-CoA. So pyruvate changes to acetyl-CoA. CoA means coenzyme A. That's where it comes from. Now we start the Krebs cycle, which, let me show you a picture of the Krebs cycle. It's a big circle. Okay, here's acetyl-CoA coming in. The CoA, coenzyme A, is going to, after it causes the acetyl group to combine with this 4-carbon molecule, the coenzyme is going to break off and do it again. So this is just an enzyme that causes this chemical reaction to occur. Okay, so that combines with a 4-carbon, making a 6-carbon molecule. This is the only one I know. This is citric acid or citrate. The rest of them, it doesn't matter if you know the names of the chemicals or the enzymes involved, except coenzyme A. That's a good one to know. All the rest of them aren't that important to know the names of. So I've got a 6-carbon molecule. I'm going to break off a carbon dioxide, and then NAD is converted to NADH. That's the same thing that happened in transition, remember? CO2, NAD, NADH. CO2, NAD turns to NADH. Now, because I lost a carbon dioxide, each of these gray balls is a carbon, I only have 5 carbons left. Another carbon dioxide is going to come off. NAD turns to NADH. Well, I lost a carbon dioxide, so I have 4 carbons left. This time, I'm going to also give off some energy. So ATP or ADP plus P is going to turn into ATP. I'm going to store some energy. Still have a 4-carbon left. A new molecule comes in. FAD is going to come in and steal 2 hydrogens. Nothing happened to carbon dioxide, so I still have 4 carbons. Still have 4 carbons. Last thing, an NAD is going to come in and turn to NADH. I still have 4 carbons, and then it's going to do it again. Now, I had 2 pyruvates, so this actually has to happen twice. So, my net gain, 4 carbon dioxides, 2 ATPs, 6 NADHs, and 2 FADH2s. That's what I gained, because I had to do this thing twice. Okay, so that just explained that. This is going to go through just what I said. You can read these, stop the slide. Actually, I'll go slower. Let's go back. Okay, that's where I was at. Let's go slower. You can stop these and read what I just told you. You can stop the PowerPoint or the video whenever you want. Okay, at the end of that, we're going to now go to the final stage, which is the electron transport chain, which occurs on the cristae membrane inside the mitochondria. Now, the cristae membrane was this wiggly, squiggly membrane inside the mitochondria. Embedded in that membrane are all of these purple molecules. These are enzymes. These are proteins. There's a whole series of them. Okay? NADH is going to drop off its hydrogen and electrons at the top of the series. FADH is going to drop it off partway down. As a result, we're not going to make as much energy from FADH as we do from NADH because it doesn't start at the very beginning. But let's go through the electron transport chain just to see what happens. Okay, now the NADH is going to start at the top. It's going to drop off a hydrogen, and it's going to drop off an electron. The hydrogen is going to be pumped across the membrane to the interior space. Okay, the electron is going to move from one enzyme to the next, on down. Okay, when it gets to the end of the series, that electron is going to be attracted to an oxygen atom. This is called oxidative phosphorylation because you have to have oxygen to create ATP. So, the last electron acceptor is an oxygen atom. That it's going to take that electron that came down here, and then we'll find out what happens in a minute. You're going to see it's turning to water. Well, let's find out why. Okay, the hydrogen was dropped off. The hydrogen's now on the interior of this membrane. And there's so much hydrogen on the inside of this membrane, it builds up kind of like water behind a dam. The only place hydrogen can come back through is through another enzyme called ATP synthase. ATP synthase builds ATP. It synthesizes ATP. If it ends in ASE, it means an enzyme, and it tells you at the beginning what it's going to do. Synthesizes ATP. So as the hydrogen comes through ATP synthase, it creates energy. That energy is then stored from ADP plus P, grabs the energy and becomes ATP. Now that hydrogen then combines with the oxygen, forming water. So the oxygen was the last electron acceptor, the hydrogen came through, we formed the water, and that's the end of the electron transport chain. That's the whole purpose of it. And that's what I just said. Okay, so how much energy did we form? Let's look at our net results. I told you 38 ATPs, so let's see how it happens. During glycolysis, my net ATP was 2 ATPs, because I remember I had to kick in 2 to get the thing started. I made 4, so my net gain was 2. I also created 2 NADHs. Well, an NADH is going to come back through the electron transport chain. Each NADH, as it goes through the electron transport chain, is going to make 3 ATP molecules. So 3 times 2 is going to be 6. I'm going to be making 6 from just those 2 NADHs. Let's continue through. You go through transition. During transition, I'm going to make 2 NADHs. Didn't make any ATP yet, but those have to come back over here. Okay, the acetyl-CoA is going to come into the Krebs cycle. During the Krebs cycle, I'm going to make 6 NADHs and 2 FADHs. I'm also going to make 2 ATPs. Okay, so excuse me, I've made 2. Now let's go back here. Depending on how this comes through the membrane, sometimes you use a little bit of energy, sometimes you make a little energy. It just depends on the electrons. Don't worry about it, but we'll just stay with what we got so far. Okay, so then all of these NADHs, here's 2 plus 2 and 6 more is 10. Each one goes through the electron transport chain, 10 times 3, because each one makes 3 ATPs, that makes 30. The 2 FADs come through, and because they don't start at the top of the electron transport chain, they only make 2 ATPs. 2 times 2 is 4. So 4 and 30 is 34. Add it all up, you've got about, the best you can do is 38 ATPs. Okay, that's the very, very best. That's where all the numbers come from. Let's continue on. Well, sometimes organisms don't have oxygen present. They still need to make ATP. But if there's no oxygen, you can't run the electron transport chain. And if the electron transport chain shuts down, then the Krebs cycle shuts down. So we've got to figure out, the organisms have to figure out a way to still make energy without using the mitochondria. So they've created a pathway called fermentation. There are 2 types, alcoholic fermentation and lactic acid fermentation. So let's take a look at both. Yeast cells, we as humans have taken yeast cells and taken a process that they normally do and kind of use them for it to make alcohol. This is what's used in winemaking and stuff like that. Okay, so what happens? The yeast cell takes glucose. It goes through and creates 2 NADHs, and it makes 2 ATPs during glycolysis, which is great. The net gains 2 ATPs. There's a little bit of energy there. But if we stop there, it can't go through. It does have a mitochondria, but it can't use the mitochondria when oxygen is gone. So in a loaf of bread, the yeast is working inside there, and it runs out of oxygen because the bread is sticky and there isn't any more oxygen. Or in the case of winemaking, when you cork the wine, it runs out of oxygen in the bottle. Okay, so once the oxygen is gone, actually even glycolysis would stop, but it doesn't, because what the yeast does is then go through alcoholic fermentation, which is this down here. It takes pyruvate, converts NADH back to NAD. The byproduct is alcohol, which the yeast really doesn't want, but the alcohol, well, leave the alcohol. The whole purpose it does it is to create the NAD, or the NAD. Then the NAD can go through glycolysis and make 2 more ATPs. It wants this top part. It really doesn't care about the alcohol, but alcohol is produced. Okay, it's kind of a byproduct. So we do it beer, wine, and bread making. Lactic acid fermentation occurs in muscle cells. Have you ever been running and get that stitch in your side? The reason you do is because those particular muscle cells in your side, run out of oxygen, but they still need ATP. And so they go through lactic acid fermentation to produce 2 ATPs. It's better than nothing, but it's not as good as 38. So almost the exact same process. Glycolysis, glucose goes through glycolysis, produces 2 ATP, it produces the NADH. They take the pyruvate, convert it to lactic acid or lactate, but by doing so, it creates the NAD. That can then go through glycolysis again and make more ATP. So they're getting 2 ATPs, but they don't get the 38. However, in muscle cells, when you stop running, you start getting more oxygen to your cells. Your muscle cells can take the lactic acid, reconvert it back to pyruvate, and then go through glycolysis. So, they could still do it. Okay, that's the end for tonight. Have a wonderful night.